Thank you. Thank you, sir.
Before double. Baby, na bada kaka. Na teaching them. What teaching them? Now teaching them. Teaching them. Some of this na me. So could I get me?
All right. Sorry, you had a network issue, and after that, I have to attend some people. So I check your pardon, Rachel.
Fine.
Rachel. Okay, you are not with me. I will have the class recorded. I already explained what a central dogma is. They started explaining what DNA replication is. The process through which a DNA molecule is, or a duplicate copy of DNA molecule is formed. And I said the process starts with the binding of a set of proteins called helicases. Binding of protein causes unwinding of the parental double strand, the DNA. So the double strand helix will be unwound to form a double strand, and the double strand also separated to form a single strand. Now, the single strand DNA have the potential of also rehybridizing again. So rehybridizing of the DNA double strand can prevent the process of this DNA replication. Therefore, another set of proteins we call single strand binding proteins will bind to stabilize, thereby preventing the single strand to come back again and anneal or hybridize. When that happens, an enzyme called DNA polymerase will bind to the leading and both the leading and the lagging strand. And I told you there is a difference with regard to how the DNA polymerase bind to the leading and the lagging strand. I explained how the binding on the leading strand leads to a continuous non-stop synthesis of the DNA from the 5' to the 3' direction. While in the case of the lagging strand, we said the DNA, because the synthesis has to proceed from the 5' to the 3', then it will start and then stop, start and then stop at a point, and then you have the formation of Okazaki fragment. So this Okazaki fragment later on will now be sealed off. So the synthesis of DNA in a lagging strand is more or less a discontinuous process, leading to formation of what we call Okazaki fragment. And after that, the Okazaki fragment will now be sealed off by DNA ligase. So this is basically what you're supposed to know about replication, a process through which a copy of the DNA is produced, the process called replication, and the replication starts by unwinding of the DNA double helix. It's remove the helix first by the DNA helicase, and then the double strand to form a single strand. And the single strand is stabilized by single strand binding proteins. And then we have the binding of DNA polymerase to both the leading and the lagging strand. We said the synthesis of the DNA in the leading strand is continuous from 5' to 3', while that of the lagging strand is discontinuous, leading to formation of a fragment called Okazaki fragments. And these Okazaki fragments are there of later linked off by an enzyme called DNA ligase. While in the case of DNA transcription, we said the process through which an information is copied from DNA to RNA. So it depends, like we said, if that information is needed for protein synthesis. So a messenger RNA is synthesized, and the RNA polymerase that is involved is RNA polymerase 2. But if what you are producing is a ribosomal RNA, and we told you what is ribosomal RNA, then RNA polymerase 1 is involved. But if what you are producing is transfer RNA, RNA polymerase 3 is involved. So in this case, in DNA transcription, a particular segment is needed or carried a particular information, for example, a particular protein. That point on the DNA will be opened off. The DNA around that region will lose the helical structure and also the separation. Pardon me. Hello. Hello. Hello? Now. Gan number ne yin ko but we back on the na ce ki bari ni yanzu nan zai. No.
Are they on?
So, as I was saying, in DNA transcription, an information is transferred from the DNA to RNA. Depending on the information, if the information needed is for the synthesis of a protein, then a messenger RNA is produced. And that process is catalyzed by the RNA polymerase II. But if a ribosomal RNA is needed, I've already explained what ribosomal RNA is, then RNA polymerase I is involved. But in the case of transfer RNA, RNA polymerase III is involved. The most important thing you need to note here is that an information is copied from DNA and then transfer that information to RNA. Either it's in ribosomal RNA, and we mentioned the function of ribosomal RNA, transfer RNA, or a messenger RNA that will be utilized for the synthesis of protein. And lastly, in the case of translation, we know what is messenger RNA. When the messenger RNA comes with the information as copied from the DNA for protein synthesis, we said it binds to a machine, a machinery, a protein synthesizing machinery called ribosomal RNA. Here in this structure, you can see this as the messenger RNA with the 5 prime, the 3 prime. Now attached to this whole molecule, which is the messenger RNA. And this messenger RNA contains these portions, and we have this transfer RNA. We say that transfer RNA brings an amino acid. So you can see the transfer RNA now attaching an amino acid. It will come and bind and then transfer the amino acid to form through the formation of peptide bond. So when this amino acid comes and attach here, it will release this amino acid to here, so it will be attached here. And then the amino acid, the transfer RNA will be released. And then another one will come, bind, attach the amino acids, and then release. Like this, until when you have a very long peptide chain. Just as we have here, you can see from the beginning, we have the ribosomal RNA here, we have the messenger RNA. As the ribosomal RNA is moving along the messenger RNA and reading the information, then the transfer RNA is coming, giving, donating the amino acids, and we can see the peptide is increasing. The length of the peptide is increasing like this, until it reaches end, where you have all the information copied, and then it is used to synthesize the protein. And you can see the protein here, the long chain of peptide chain, which can then be folded and then to produce a functional protein. This is basically translation. We say translation is the process through which the information on the messenger RNA is used to produce a protein. I already explained what is RNA naturation and denaturation. I say the process through which the RNA will lose the double helix, and at the same time also the double strand will become a single strand, or will be separated to form a single strand. And we said so many factors, which include the temperature, which include the pH, but it could be the acidic pH, or it could be the alkaline pH, or ionic strengths, but actually cause this separation in the DNA strand. And the next thing which I said I will explain after this, the functions of the nucleotide, synthetic nucleotide. In clinical medicine, because synthetic nucleotide can be used in different areas, but I will discuss on something that is a bit more relevant to you. In clinical medicine, we know that synthetic nucleotide has been used as a therapeutic agent, most especially in the treatment of quite a number of disease conditions, like most commonly viral disease, viral infection, and as an anti-cancer agent. And what happened is mostly these substances are used to either inhibit some of those processes we just mentioned, either the process of protein synthesis the process of DNA synthesis or the process of RNA synthesis. For DNA synthesis, it's called replication. For RNA synthesis, we call transcription. And for protein synthesis, it's called translation. So how these substances can prevent DNA synthesis? We know in DNA synthesis, what actually happens is when the DNA polymerase binds to the DNA strand, the nucleotides will come and then they will be joined by this formation of phosphodiester bond by this DNA polymerase. But in a situation whereby an individual was given one of these synthetic nucleotides, these synthetic nucleotides, they are like nucleotides, but they are not exactly like nucleotides. They tend to be more reactive than the nucleotides, a modified form of the nucleotides. So once this DNA polymerase pick one of those synthetic nucleotides, because of the reactivity, so it will block the active site of this DNA polymerase. So the DNA polymerase can no longer synthesize the protein. I remember, sorry, can no longer synthesize the DNA. Or in the case of transcription, if it is transcription, the RNA polymerase will no longer be able to synthesize the RNA. So in this classical case of cancer, let's say, it's uncontrolled cell division. Cell will continue to divide. So it divide. But in a situation whereby for a cell to divide, there must be a complete duplicate copy of the DNA. So if the synthesis of the DNA is being blocked, the cell can no longer divide. Therefore, the cancer cells will die. So this is how the proliferation or the uncontrolled cell division in cancer cells is being blocked through these chemicals that has been used as drugs. And also in the case of antiviral, where you have the viral genome being integrated into a genome of the individual that was infected, and then this viral genome is being transcribed to produce viral proteins. So if this synthetic nucleotide binds to the RNA polymerase, it will prevent the synthesis of these viral proteins that tend to be infectious or that causes all these damages that with quite a number of viruses. There are quite a number of drugs like cytarabine, RC, which is used for the treatment of leukemia. This cytarabine actually inhibits DNA polymerase. That is the mechanism of action. And there is also pifluorouracil, which is a very well-known anti-cancer agent. It also inhibits DNA polymerase. There are quite a number of also antiviral drugs like remdesivir for the treatment of COVID-19 Ebola, which is an analog of synthetic analog of adenosine. It also inhibits DNA polymerase, like acyclovir, which is a guanosine analog. It also inhibits DNA polymerase used for the treatment of herpes simplex virus, the simplex virus. And we also have zidobudin, which is a thymidine analog, which inhibits reverse transcriptase. So this zidobudin is used in the treatment of HIV infection. And like I said, it is a thymidine analog. It inhibits reverse transcriptase. Also, a similar drug, which is lamivudine, is also a structural analog of cytidine analog. So an enzyme, the RNA reverse transcriptase, will now pick this lamivudine, thinking that it is cytidine, in order to synthesize a DNA. So, but in order to synthesize a DNA from viral RNA by an enzyme reverse transcriptase, in the process, it will now pick this lamivudine. And in picking this lamivudine will bind to the enzyme active site, reverse transcriptase, and then block the enzyme so that the DNA will not be produced from the viral RNA. As such, the infection will not continue. So these are basically some of, because there are many you can search, and you find out quite a number of those drugs that some of them you are very much familiar with, and this is actually their mechanism of action. If you have any question, please you can ask. I can see the only person that is here is actually not with me, is doing some distance is not with me. So please, you can watch the video, and then if you have any question, you can ask. Thank you very much.